So these pictures show you our research area. It's a shallow peat lake called the Leijen um, in the north of the Netherlands. It's about 1 meter 20 deep, and it has extensive reed beds surrounding the lake, as you can see here. Behind the reed beds, you see the agricultural fields, grasslands, meadows, hay fields. You see there's quite a bit of inlets of shallow water between the reedlands, and there are some islands in this lake. Lake is used for recreational boating. You see some floating leaf plants here, but there's actually no submerged aquatic plants here, which is the part of the main ecological problem in this lake. So here we're zooming into a recreational home that's only used in summer. And here's a small beach where people can go swimming. And then here's a harbor for some recreational boats. And this is where the CANAPE project for VHL is conducting its field research. The lake is nearly perfectly circular, 3 kilometers in diameter. Welcome all. My name is Jasper van Belle. I'm a eco-hydrological researcher at the Van Hal Larenstein University of Applied Sciences, or in brief, VHL. We're a professional education institute and research institute in the north of the Netherlands. So we're conducting lake ecological research within CANAPE. We're studying the effects of Bream on water quality and in a shallow peat lake. Goal of this all is to figure out if we can find new ways to restore the aquatic ecosystem to its clear water state.
So I'm Harry Mack. I'm the project manager for the overall CANAPE project based in the UK. If you've watched any of our other videos, you'll have seen the wider CANAPE project, and I encourage you to go and watch them. We're also joined today by Tsjippe and Joop.
Hello, I'm Tsjippe Visser. I'm a researcher at VHL University. And within the CANAPE project, I've been mainly responsible for the water quality measurements.
My name is Joop van Eerbeek, and I'm a fish migration biologist, and I work on spatial distribution of bream within the Lake Leijen for the CANAPE project. So we are mainly looking at bream as a bioturbator within the lake.
Thank you, everyone. Start with Joop. Joop, do you want to explain what this fish is on the left?
Yep. So on the left we see a common bream, which is one of the most common fish in the Netherlands. Its Latin name is Abramis brama. They can grow quite large, up to 75 centimeters. They're always colored between bronze and this gray, depending on the type of lake. The darker the lake is, the more silvery the fish will become. So, and bream, they root in the bottom, and in this way they bioturbate the lake. So they make the lake a bit more turbid. Small bream, they are filter feeding, eat on phytoplanktons and zooplanktons within the lake. And as they become larger, they start to eat larger prey. So in the end, they will eat snails and mussels and all sorts of things living in the bottom. But by foraging around, they uproot the bottom and they cause a lot of murkiness in the water.
Okay, and with the picture on the right, what have you been doing with the bream?
We've been tagging Bream. We've been using 2 telemetry techniques, and on the right we see pit tagging, which is a quite cheap way of tagging fish. So pit tags, we inject a transponder in the fish with quite large needles we see on the far right, the red ones. The pit tags are sort of standalone. They don't emit signals, but as a fish passes through an antenna station, the tag will be triggered and it gives off a unique number, and in this way we can follow the fish through our pit tagging stations.
And so if we jump to our next image, you can see some of the tracking you've been doing here. So I'm guessing on the right is one of your receiving posts. Can you talk us through what these posts are doing and how they're working with the tags?
So as you can see on the left picture, we had 28 stations stationed within the lake. On each of those posts, there's a hydrophone, a VEMCO receiver, and as the fish pass by the VEMCO receivers, we get a triangulation. So they're picked up by multiple stations at the same time, and that's what the picture on the left illustrates. The darker green the shading is, the better our reception is. So only in the red parts we don't pick up the fish, but in the rest of the lake we get a triangulation point, and we know exactly up to 10 centimeters where the fish is swimming. Even height we can measure, so the height in the water. And that gives off quite cool pictures. So this is a little movie of one brim over 2 weeks swimming through the lake, foraging around. And the fish is always in the front side of the scatter. But it's picked up by multiple stations, so that's why the scatter looks a bit distorted, but it's always in the front. What we found out is that in the southwestern corner is the soil type is a little bit more sandy, so the water is a little bit clearer, and brim tend to forage a little bit more in that area because the sandy soil type, I think there's a bit more availability of Dreissena mussels, of these tiny little mussels which they are foraging on. But that is just gut feeling. We still have to look in that what exactly he's been doing there.
And Tsjippe, do you want to come in now and explain what you have been doing with your water monitoring research?
Yes, of course. So next to the fish monitoring, we've also been monitoring the water quality. And here you see two of our students busy on the lake. They're actually having a little break here, calibrating all the sensors. And on the next picture, you can see our monitoring route. And we use two sensor units to monitor around the lake. We use a Hydrogen 10 sensor, which takes in situ measurements every 30 seconds whilst we navigate around the lake. And this sensor contains sensors for measuring the pH, the temperature, electroconductivity, calcium, chloride, sodium, potassium, ammonium, nitrate, and it also has a reference sensor. And then we have another sensor unit, the HADA multimeter, with which we measure or monitor the turbidity and the dissolved oxygen. And we have these sensors placed on two little surfing boards, as you can see on the picture on the right. So we take up the water from a depth of 30 centimeters and then pump it into the boat. You can see this on the picture on the left, where the hydrogen sensor is in the water that's been pumped up, and it's linked to a tablet with which we registrate all the different parameters every 30 seconds. And under the tablet, you can see the sensor unit with the different sensors. And then here we have a graph of the nitrate concentrations, but I think Jasper could better explain this.
Yeah, so what we see here is total inorganic nitrogen as measured with a Hydrion setup and interpolated between measuring locations. So this is nitrate plus ammonium for those interested. So this is bioavailable nitrogen in the water column. Green is low levels and red is high levels, which in this case are pretty high levels. So this shows that the water quality can be very variable in space, even in such a simple lake as Leijen. Large variation within the lake, and well, this is important because the regional water authorities, they have one monitoring station in the center of the lake used for the Water Framework Directive reporting. So, well, this points to well, maybe we should reconsider whether one measuring point in the middle of a lake is all that representative of the water quality of a lake. Um, so there's quite a bit of variation. And if we go to turbidity, we see that there as well. There's a lot of variation in turbidity from very clear in the northeast at this date. The patterns change very much day by day. But on this day in the northeast, it's very clear water, and in the southwest we see rather murky water. And this is where we've done some extra investigations, and that shows us that it's mostly suspended solids. So it's the lake sediments that being churned up and that's moving around in the water column that is causing the turbidity. Algae growth as a minor contribution to it. So if we can go to the last slide, you see that this is where the bream are at. So this is for spring, summer, autumn, winter for 10 different individuals. The red places is where they are, well, have been recorded most. And you see that there's not that much variation between the seasons, but you see that the southwest corner is one of the places where they really like to be. It's one of the preferred places for these bream, together with these spots in the center of the lake. And if you then recall that the turbidity is highest also in the southwest corner, and this is a repeated pattern, then we show that the habitat preference of bream within the lake affects where the lake is most turbid. And this is very important for our goal to restore the lake to a clear water state, which is dominated by submerged water plants. These submerged water plants need sufficient light to grow, so they grow best when the water is clear. And obviously, the best locations for this are away from where the bream are at. So this is important information for the regional water authorities to base their restoration plans upon. And that's the goal of this research.
So what is the next step with this research? Is it already being used by the regional water authority, or?
Well, one of the things is that we've shown this variation in space, and well, this of course got them thinking how to deal with this. But it's not that easy. You don't change the central setup of monitoring for what framework directive reporting. But it is well, it's an important thing to keep in mind. And actually, the amount of spatial variation that we found is a bit larger than we expected. So that's food for thought. And this, well, it's just very fresh finds. This, so there's no concrete action being tied to this. But one of the things that we're looking into is if you can sample the animals, the macrofauna living lake bottom in the sediment, and use this as a proxy for where the bream like to be. So if you can, it looks like if you can determine where the food for the bream are, then you know where the bream are. So then in future, if you want to do a restoration project, you can just do a one-time sampling of the sediment macrofauna, and then place the restoration effort away from the preferred bream spaces.
And is that because the bream disrupt the planting of new water plants?
Well, the bream, as our research has shown, the bream increased turbidity, thereby decreasing the survival chance for submerged macrophytes. So anything you do to promote these submerged plants, submerged macrophytes are just plants growing under the water. So you want to try and enhance these away from where these bream are churning up the bottom and making the water very turbid, and actually also uprooting your plants if they start to grow. So those are the places with lowest chance of success if you want to do restoration measures, and the places where the bream stay away from, those are the best places for your restoration effort. So it helps to increase restoration success